Hello everyone, welcome to the new session of Organization of the Genetic Material. So in this session, we are going to study about the organization of the genetic material in the case of prokaryotes. Okay, so DNA must be replicated when the cell is ready to divide. So when the cell is ready to divide, the DNA must go for the replication. So what is replication? That is the copy of DNA, making copy of DNA. That is called as replication, or it is also called as duplication of DNA, right? So it must be read to produce the molecules such as proteins to carry out the functions of the cell, right? So DNA must be read, right, to produce the molecules such as, so it produces different types of proteins to carry out the functions of the cell. So in this reason, DNA is protected. Okay, DNA is protected. It is organized and packaged in a very definite and a specific ways, right? So that is why it is called as DNA is protected and packaged in a very specific ways. In addition, DNA molecules can be very long. So DNA molecules are very long. In the case of human, it is around 2 meter long. 2 meter long. Okay, so it is stretched end to end. The DNA molecules in a single human cell would come to be length of around 2 meters. Just now we discussed about, and you can now imagine the cell which we could not see with our naked eye, right? Suppose this is an eukaryotic cell. In fact, it is a human cell and it is having a nucleus. So we cannot see the cell, we cannot see the nucleus, and the chromatin material which is present inside the nucleus is 2 meter long in the case of human cell. So now you can understand that the cell which we could not see with our naked eye, it is having 2 meter long chromatin material in its nucleus. So you can imagine how much it is organized, how much it is packaged and present inside the nucleus. Okay, so the DNA for a cell must be packaged in a very ordered way to fit and function within a structure, the cell that is not visible to our naked eye. Majority of an organism genome is organized into the cell's chromosome, right? So normally the cell is having a nucleus and it is having chromatin material, but when the cell is going to undergo division, the chromatin material divided into different fragments, and those are called as the chromosomes. So actually it is a genetic material, whether it is present in the case of chromatin material or in the form of chromosomes, which are a discrete DNA structures within the cell that controls cellular activity. So all the cellular activity is controlled by this DNA structure, because from the DNA, RNA are synthesized, and from RNA different proteins are synthesized. Now these proteins are very, very important for the formation of structure, right? For biochemical reaction going under the cells. Okay, so all these are involved, like for this protein is involved in all structure as well as the biochemical reactions of the cell. Now how these proteins are formed, now you can understand. The main thing is DNA, right? And that is present inside the nucleus, and it is very, very much organized. Okay, so eukaryotic chromosomes are housed in the membrane-bound nucleus that is surrounded by the nuclear membrane. So this eukaryotic cells are quite organized. All the nucleic material, all the nuclear material is present inside the nucleus, and nucleus is a membrane-bound organelle, and membrane is called as the nuclear membrane. Nuclear membrane is having nuclear pore. Okay, that is very, very important, because the DNA is present inside the nucleus, and from DNA RNA gets synthesized. Now synthesis of protein synthesized outside this nucleus. So this RNA has to pass outside the nucleus. From this pore, from these pore, this RNA comes out. Okay. So this is very, very important. Now, the liquid material which is present inside the nucleus, that is the nucleoplasm. Okay. And it is having a chromatin material inside it. Okay, it is having one more round structure which is called as nucleolus, and it is a site where the RNA formations takes place. So we will study more about the eukaryotic chromosomes organization in one more separate session. So here we are going to focus more on the prokaryotic organization of DNA. Okay, so most prokaryotes contain a single circular chromosome that is found in an area in the cytoplasm called as nucleoid. So we can see here, this is a bacterial cell, this is a prokaryotic cell, and it is having a nucleoid structure. The chromosome is in the form of circular, right? Circular single chromosome structure. Right? Okay, but it is not bound with any membrane like structure. It is not. And this chromosome, this single chromosome is present in the cytoplasm itself. And this particular structure is called as nucleoid. It is called as nucleoid. The nucleoid mean nucleus-like. It is not a true nucleus, so it is called as nucleus-like. Is an irregularly shaped region within the cells of a prokaryote that contain all or most of the genetic material. So all the genetic material is present here, but beside these, some circular, small circular DNA material is present. Okay, that is called as plasmids. But the main DNA is this single circular chromosome which is called as nucleoid. This is the main DNA structure in the case of prokaryote. The length of the genome widely varies, but generally is at least a few million base pair. Okay, it varies from one prokaryote cell to the another, like one bacterial cell to the another, but generally they have a few million base pairs. Okay. The term chromosome comes from the Greek word for the color chroma, because when they get stained, right, they impart some color, and body soma because they are cell structure, because they are having some structure, so that is why it is called as soma. Okay, so that are strongly stained by some colorful dyes used in the research. So to know where this chromosome structure exists, some stains are used, some dyes are used, right, to see the structure of the chromosome. Right, so that is why it's got its name, because it's show a particular color. Okay, and it is having a structure, like that is called soma. So combinedly it is called as chromosome. Each chromosome is made up of protein and a single molecule of deoxyribonucleic acid, that is DNA. So it is made up of DNA and some protein structure. The size of the genome in one of the most well-studied prokaryote, that is E. coli or Escherichia coli, that is 4.6 million base pair, which found extended a distance of about 1.6 millimeter. It's stretched out. So when we stretch out of a human DNA, then it is around 2 meters, and when you stretch out E. coli bacteria, it is 1.6 millimeter. Now you can compare how small it is, right? But still, it is an entity. It is a one species, that is a species of a bacteria, that is E. coli, and it is having 4.6 millions of base pairs. Okay. Okay, so the DNA is twisted beyond the double helix in what is known as supercoiling. This is very, very important. The coiling, and it gets supercoiled. So, in a very small space, this DNA has to be placed and organized, right? So it has to undergo coiling as well as supercoiling, right? So here you can see the supercoiled domain structure. Okay. Some proteins are known to be involved in the supercoiling. Other proteins and enzymes help in maintaining the supercoiled structure. So there are some specific proteins that are involved in the supercoiling. Okay, beside these protein, there are many enzymes that helps in maintaining that supercoiled structure. Okay, so one of the example is this topoisomerases. This is an enzyme, and when you will study about the replication of DNA, duplication of DNA, you will come to know more about this particular enzyme. Okay, so topoisomerase are involved in the supercoiling of the DNA. DNA gyrase, it is a type of topoisomerase found in bacteria and some archaea. Archaebacteria are also known as archaea, that helps prevent the overwinding of the DNA. So these two enzymes are very, very important for the supercoiling of the DNA structure. Some antibiotics kill bacteria by targeting the DNA gyrase. So when this DNA gyrase is destroyed by this antibiotics, the bacteria get killed. So how important these enzymes, this DNA gyrase is, now you can understand. If this enzyme is absent, DNA gyrase, the bacteria can get killed, right? So, in addition, histone-like proteins, in the case of prokaryotes, this histone is absent. This histone is not present, right? But histone-like proteins are absent, that binds and aid the DNA packaging. This histone protein is present. Histone protein is present in case of eukaryotes. It is present in eukaryotes, right? But in the case of prokaryotes, histone-like protein are present, right? That are called as polyamines. So histone-like proteins, not histone. Other proteins bind to the region of replication, the location of the chromosome where the DNA replication initiates. Okay, so some other proteins are also involved in the packaging of the DNA. Some proteins are also involved, they get attached to the origin of replication, okay, that is located on the chromosome where the DNA has to initiate its replication. Okay. Chromosome in bacteria and archaea are usually circular and typically contain only single chromosome within the nucleoid. Only this is the single chromosome present inside the nucleoid. Because the chromosome contain only one copy of each gene, prokaryotes are haploid. Like we are having each gene is present in the two copy, okay, a pair, in the form of pair, but here single chromosome is present, so all the genes are present as a single copy. Okay, this is very, very important, and that is why prokaryotes are haploid in nature. Because different regions of the DNA are packaged differently. Some regions of the chromosomal DNA are more accessible to the enzyme and thus may be used more readily as a template for the gene expression. Okay, so there are some chromosomal DNA that are more accessible to enzyme, means the enzyme can get attached to that particular part or the portion of DNA very easily, right? So that may be used more readily as a template for the gene expression, means that part of a DNA is readily involved in the gene expression. So that particular part is open, quite open. Okay. The nucleoid is composed of DNA in association with the number of DNA binding protein. So that is histone like protein, but again I'm telling, it is not a histone. It is histone like protein that help in maintain its structure. Organization of the prokaryotic chromosome. So what are the things that are involved in the nucleoid structure? So the isolated nucleoid contain 80% of DNA. Okay, so nucleoid is having 80% of DNA structure, 10% of protein and 10% of RNA structure by weight. Okay, so if we talk about this protein structure, that's 10%, there are 4 major families of bacterial histone like protein. Okay, so what are those? Histone like nucleoid structure protein, HNS, histone like nucleoid structuring protein, heat unstable protein, factor for the inversion stimulations, and integration host factors. So these are the four families of protein involved in the organization of the nucleoid structure in the case of prokaryotes. So coming to the first one, histone like nucleoid structuring protein, HNS, it belongs to the family of the bacterial protein that play a role in the formation of nucleoid structure. So they are mostly involved for the structural organization of the nucleoid. It is believed that HNS achieves this by forming complex with its self and binding to the different sections of the DNA. So they are more like histones. Okay, histone also get organized in such a way and it is binded with the DNA structure. So this histone like nucleoid structuring protein are also bind to the different sections of the DNA. So they are more of a structural organization type of protein. Okay, next family of the protein is the heat unstable protein. Okay, so this protein binds non-specifically to the DNA and bends it. The DNA apparently wrapped around the histone unstable protein. So they are not specifically bound because they are not involved for any structural organization of the DNA, right? So they are present at the bends and apparently wrapping around the HU protein. Next is the factor for the inversion stimulation. What is this? It stimulates DNA related process such as DNA inversion and excision. When the DNA has to cut, this particular protein is involved. That is excision. It activates transcription of tRNA and rRNA genes and regulates its own synthesis. So how the DNA is involved in the formation of RNA that later transformed into tRNA and rRNA. So they are mainly concerned for this particular excision and activities. Integration of the host factor. It facilitates bending of DNA by binding the specific DNA sites. Okay, so this is all about this particular session that we discussed in this session about the organization of prokaryotic chromosome. Okay, so in the next session, we are going to study about the organization of eukaryotic chromosome. Okay. So till then, thank you very much.